All right, so thanks for joining us today. We're very excited to have Dr. Jennifer Estell presenting today. Jennifer was the recipient of the 2020 Young Scientist Award, so we're very lucky to get this presentation today. If you have questions, there's a Q&A box down at the bottom of your screen. You should be able to see it. You can type your questions in there, and we'll have a Q&A session at the end of the presentation. So yeah, we'll get started. So I'll pass over to, again, our 2020 Young Scientist Award winner, Dr. Jennifer Estell.
Hi, everyone. Thanks for coming. Thanks, Grace. I really appreciate the invitation and, of course, the honor of receiving this award. Just to let everybody know, if you want to keep up with us on Twitter, my handle is at DNA Chicken, or our website, which is diabetesresearch.ca. I'm going to move forward. Oh, and for those of you who aren't familiar with us, with the ARGCIA in Montreal, which is a small research institute that's affiliated with the University of Montreal and McGill. And I've been there, I'm now an associate research professor. I've been there for 9.5 years now, so coming up on the 10-year anniversary. So when I was putting together this acknowledgement slide, I wanted to, show it first, because I often get to the end and I'm in, a rush to keep time, and I don't really give the proper attention to all the people that were involved in really, getting this award. Really made me go back and take stock of really all the people who have been involved in this work, either in, large part or a small part, but everybody listed here has contributed something to the work that our lab has produced over the last almost 10 years, and it's just kind of humbling and a bit, you feel a bit proud to just see all of the people that have been involved. The team that I have, that was last year, a picture we took while we were curling. We're much smaller this year, but it's great to see all the names of the people that I interact and still interact with, and we still have many ongoing projects. I also wanted to highlight again that Diabetes Canada was the first grant that we received in the lab as a young lab, as assistant research professor. So, it's a big thanks to Diabetes Canada for really being the first to fund us. Much of the work that I'm going to present today was funded through that grant and that initiative, and has led to a lot of the work that we continue to do in the lab. So we're really interested in diabetes, of course, but really in this population of people that don't yet know they have diabetes. So early on in the disease, because we're interested in this early kind of stage and what's going wrong, we spend a lot of time just trying to figure out how our system, our body, energy metabolism just works normally, normal physiology, normal molecular biology, and the mechanisms in which our tissues talk to each other, enable to regulate glucose levels and glucose homeostasis. We've focused a lot in recent years, if you have followed our work, on fatty liver disease and its role in diabetes. We have focused a lot in the past on the beta cell. We really kind of take an integrated approach of how these two organ systems interact together, but we also think about all the other organ systems that are involved, the muscle, the adipose tissue, as well as our projects have led us in a very different way that I didn't originally think of going in the beginning. Through collaborators, we've kind of gone into looking at cancer and how cancer progression and prognosis is related to metabolic status, and how having diabetes increases your risk of developing cancer. So, it's kind of taken us in a lot of different directions. But what I want to do focus today, in kind of honor of this award and to think back, is kind of start and tell a story of how it developed from the beginning, and how my original interest in understanding liver metabolism and really how gluconeogenesis was regulated was what started off, and what inspired a lot of the work we did, and how that took us in so many different ways, but then eventually dialed back again to looking at gluconeogenesis. So it's kind of the work I'll present is really an ongoing story that probably spans about 15 years of how I've been thinking. So it originally started a long time ago, where we were interested in figuring out what are really the underlying causes of diabetes or metabolic syndrome. What is going wrong in the tissues at the molecular level that eventually leads to that tissue not being able to regulate glucose properly in the blood? There are many many hypotheses out there, there's a lot of evidence that many pathways are involved. Not one of them is not more important than the other. We've really honed in on the role of mitochondria and what maybe used to be called mitochondrial dysfunction. I think that term is not used as often anymore, and how that fits into this. But we also, we also always take stock of all of the other pathways that are concurrently being dysregulated in diabetes, and how they kind of cross talk with each other. So our work has taken us into, the role of meta-inflammation, ER stress, insulin resistance. We focus a lot on diets, a lot of lipid metabolism. So we touch all of these areas, but really the heart of what has fed our research and our interests over the years has been the role of mitochondria and what they do. And this figure, although the text is really small, I think you don't really need to read it, but I think it illustrates very nicely how the mitochondria are kind of central to a lot of things in terms of regulating energy metabolism. I mean, the traditional view of saying the mitochondria is the powerhouse of the cell, it's a bit simplistic, but one of its main roles is obviously to promote ATP generation through the electron transport chain. It does this by converting energy equivalents from our diet, from glucose, from fatty acids, from other energy sources. So, this process is very important. There's a lot of evidence over the years that dysfunction of mitochondria can eventually lead to problems in regulating blood glucose levels, problems with insulin sensitivity in tissues, muscle, adipose, many tissues, even, endothelial cells. So, the mitochondria in each specific tissue seems to eventually, if you have problems with it, will lead to these problems. But what happens in between was really not understood. How does, mitochondrial dysfunction or the inability of the mitochondria to deal with stress eventually lead to these problems? What are the molecular mechanisms? So I spent a lot of time over the years thinking about this, and the mitochondria are really, subject to a lot of stress. Glucolipotoxicity, environmental toxins, they can accumulate mutations in the mitochondrial DNA. The mitochondria don't only do electron transport chain or respiration, they're also very intimately related to apoptosis. So there's really a lot going on, and it's really a very big area of study, and that keeps us busy. And what got me originally interested in whether mitochondria or problems with them was involved in how diabetes developed was, some earlier work by Mary Elizabeth Patty and Vamsi Musa, who had shown at the transcriptional level anyway, that problems with mitochondrial gene transcription, not transcription of the mitochondrial genome per se, but genes from the nucleus that are responsible for making more mitochondria or ramping up mitochondrial metabolism are all downregulated in patients that have diabetes. So it's an entire program. It's not one particular mediator, but you see a big downregulation of mitochondrial gene expression. And it wasn't just people who had diabetes. If you looked at family members that have or people with a family history of diabetes, they also seem to have downregulation of these pathways. So this provided some evidence that maybe having lower mitochondrial capacity, lower respiratory capacity could be a predisposing factor to developing diabetes when you're challenged by other things. At the time, I worked in a lab that had a lot of tools and focused a lot on this transcriptional coactivator, PGC-1 alpha. So we got the idea at the time that if we were able to modulate PGC-1 alpha activity, which is a upstream regulator of this entire program, could this help us to ask the question whether decreased mitochondrial function capacity or adaptability, does it contribute to the development of diabetes? And this kind of concept has turned maybe more recently into the term of mitochondrial inflexibility, perhaps. So we were really interested in that. And it wasn't really the role of PGC-1 alpha per se, but we were thinking of using PGC-1 alpha as a tool. Right? We know that it is a transcriptional coactivator that drives the expression of multiple transcription factors in the nucleus. And these transcription factors have, a common purpose, which is to drive the entire program of mitochondrial biogenesis, increased electron transport chain activity, increased ATP synthesis. It's also responsible for driving enzymes that will detoxify ROS. So by upregulating this pathway, you're able to upregulate mitochondrial capacity. You can increase the number of mitochondria in cells, increase their density, increase the amount of electron transport chain, units, but also help to detoxify ROS and just make a more efficient system. So this coactivator, it's a very short-lived protein. It's there to really ramp up mitochondrial activity in times when you need it. That would include when you're fasting, when you're exercising, when you have inflammatory insults. Insulin is known to regulate this pathway. So kind of any kind of metabolic stress or metabolic challenge, you'll have increased levels of PGC-1 alpha, either expression or activity. It's regulated by a number of different kinases. Now it's a highly regulated axis or protein. And it integrates the external signals of metabolic stress, metabolic challenge, in order to ramp up mitochondria when you need them. So we thought this was a really kind of cool tool to study the question, really, if you are not able to ramp up your mitochondrial metabolism, increase your respiratory capacity in times when you need it, does this eventually lead to diabetes? And, the hypothesis to use this as a model, didn't come out of nowhere, because we know that in people that are obese, people who have diabetes, metabolic syndrome, fatty liver disease, cardiovascular disease, we know that in general, they actually have decreased levels of PGC-1 alpha. There's now a number of different studies. The original studies were published, 2003, early 2000s, but now there's just abundance of evidence out there that suggests that mitochondrial or metabolic disease is associated with low levels of PGC-1 alpha. And when I write PPAR gamma coactivator 1 alpha, this is the gene, and PGC-1 alpha is the protein. There are also, now we know, common SNPs, or single nucleotide polymorphisms of PGC, that are also involved in diabetes. So there is evidence out there that not only are we modeling, using it as a model of mitochondrial inflexibility, but it's also associated with diabetes. So we wanted to know how. We decided to take a tissue-specific approach, and I don't want to really go into the background of why the whole body knockout has major neurological problems, which would interfere with our interpretation. And, we know that the muscle, fat, liver all play roles in regulating glucose metabolism. So one of our first studies was actually to look at the muscle, because the early studies which saw low PGC-1 alpha in diabetes were involved decreased expression of PGC in skeletal muscle. So we want to ask the question, actually, does low levels of PGC-1 alpha in skeletal muscle actually lead to diabetes? Concurrently with this, we knew that aging is a significant risk factor for diabetes. As you get older, your chance of diabetes increases, and which is also associated with decreased mitochondrial function and decreased mitochondrial inflexibility. So the question really is if your muscle has lower levels of PGC-1 alpha, as you're aging, does this affect the development of glucose intolerance and insulin resistance over time? So to do this, we took flox mice. We did a skeletal muscle specific knockout of PGC-1 alpha. And we had mice that were litter mates. Everything was controlled, but some were sacrificed, and studies were done at 3 months old, and some were sacrificed much, much older. So 2-year-old mice. This is a very long study. We took the muscle from these mice, and we did, that time was microarray analysis. And we compared to see really what the programs were that were affected by, 1, loss of PGC-1 alpha, so that's genotype, and 2, aging. And so we can focus, 1, on the red bars, which is loss of PGC-1 alpha. We think that this model was very much turned out to be what we intended it to be, which was a model of loss of mitochondrial function or mitochondrial inflexibility, because then almost every single pathway that came up significantly regulated by PGC is involved in mitochondria. So electron transport chain, TCA cycle, ATP biosynthesis, fatty acid oxidation. These were the top pathways. So we know that the main things that we disrupt when we disrupt PGC is the mitochondria. If you overlap this with the processes that are regulated just in aging, so independent of the phenotype, it's not very clear, but this is not the top pathway in aging. I just wanted to overlap what was the same with PGC. But electron transport chain and the other mitochondrial pathways were definitely a component of aging, which was already known. This is not a surprising. We know that this happens is that as you age, your mitochondria become less efficient. But you can see that loss of PGC had much more of a significant effect. So this assay won't tell us, the biological significance or impact. So the microarray told us definitely that we are affecting mitochondria by loss of PGC. We could confirm that at the individual gene level. So what was interesting we found is that in young mice, these are 3-month-old mice, this is just on a regular chow diet. You can see that most of the nuclear encoded genes for mitochondria are downregulated, as well as some mitochondrial encoded genes. You'll see that succinate dehydrogenase activity in the mice in their muscle was decreased. So mitochondrial respiratory capacity was just basally decreased in these mice. So we're pretty confident again that we created a model of mitochondrial respiratory insufficiency. But this was surprising to us because the idea at the time was that if mitochondria don't work properly, you would get glucose intolerance and insulin resistance. And we saw completely the opposite. So in young mice that have low levels of PGC-1 alpha in the muscle, they actually do better in terms of, this is a glucose tolerance test, than their wild type neuromate controls. So this was surprising, but at the time, there was starting to be more and more evidence that in other models of mitochondrial insufficiency, this was actually true. So young mice are fine, and actually it might be beneficial to them to inhibit mitochondrial function. If you compare this directly with the old mice, so this is 2-year-old mice, you can see that loss of PGC-1 alpha, exasperated the reduction in gene expression in terms of mitochondrial genes. It exasperated, in aging, you see decreased respiratory activity. You see that the loss of PGC-1 alpha even made that even worse. So again, we kind of made the situation worse in the mitochondria as we went on. And when we followed the same mice over the 2-year period, they started off more glucose tolerant, and they eventually ended up less glucose tolerant. So the combination of losing PGC-1 alpha and aging eventually led to the mice being significantly more glucose intolerant and insulin resistant. So we wanted to dig deeper into why that happened, actually, because originally we were looking really in the muscle, trying to figure out why the muscle was not working properly in these mice. And in the end, we figured out the muscles seemed to be working fine, not in terms of respiratory capacity, but looking at GLUT4 expression or insulin sensitivity, things like that. There wasn't much difference in the muscle. But what we started to see, which surprised us, is that if you looked at inflammatory markers in the muscle, we didn't see increased inflammation. And increased inflammation or inflammatory signals has been associated with loss of mitochondrial function in other studies, right? So there seems to be some link between having efficiently working mitochondria and activating kind of this low-grade inflammatory response. But what we did see was, while we didn't see any inflammation in the muscle, we started to see, even at the earlier stages, inflammation or higher inflammatory signals in liver and fat. So remember, this is a muscle-specific knockout of PGC-1 alpha. So somehow loss of PGC-1 alpha in the muscle was leading to a whole body systemic inflammation. And the hypothesis we were working from at that time, which I think we still kind of believe today with there's more evidence, is that the PGC-1 alpha, the mitochondrial dysfunction or inflexibility in the muscle leads to the muscle secreting factors or cross-talking with other tissues to cause dysfunction in other tissues. And it's likely the dysfunction in these other tissues. We didn't look at the pancreas in great detail, but we did see problems with the islets as well. So the fact that we had crosstalk with these tissues, and we think it's the problem with these tissues in particular that's causing the eventual development of the glucose intolerance. And around the same time, so we published that in 2014, around the same time, a number of different papers were coming out to show an abundance of secreted factors that PGC-1 alpha in muscle affects. So losing PGC-1 alpha will affect the secretion of many of these things, which affect a variety of tissues, coming together, just illustrating the fact that whether this is PGC dependent or mitochondrial dependent is still, a matter of debate, because PGC can do things other than regulate mitochondria, but it's still, I still favor the hypothesis that it really is, metabolic inflexibility in the muscle that's causing this altered systemic circulatory response, where different things are being secreted, different metabolites are being secreted, and this educates then the other tissues surrounding them. So while we had some evidence that definitely the interaction between age and loss of PGC-1 alpha in muscle could contribute to the altered glucose metabolism that we are seeing, that could be associated in humans, we wanted to dig deeper, because the role of mitochondria in islets and beta cells is obviously very important. We all know, as many of us islet folks and beta cell folks know, is that the ATP synthesis and mitochondrial function is really needed as that signal to stimulate insulin secretion from beta cells. So it's not surprising that mitochondrial dysfunction in beta cells might eventually lead to inappropriate or inefficient insulin secretion. And so there was some evidence in the late 2000s by Charlotte Ling showing that PGC-1 alpha expression in islets from people with type 2 diabetes was very much reduced. And what we found very interesting about this paper, and I'm going to touch on this a little bit later, is that there was a SNP in the PGC-1 alpha gene in humans that was also associated with lower insulin secretion. So there was evidence that possibly PGC-1 alpha could be involved in GSIS. And so we wanted to use our model in mice to kind of tease out what were the mechanisms behind this. Was it really, really just linked to ATP production, and whether this in itself as a single defect could lead to, inappropriate glucose regulation. So we created a islet or beta cell specific knockout of PGC-1 alpha and beta. We did this because PGC-1 alpha and beta are very similar, and we found in at least beta cells, for sure, other tissues as well. When you knock down one, often the other one will go up. So in this case, we really, to nail down the pathway, we wanted to knock down both to cause a very, significant effect on mitochondrial flexibility. So we made that knockout. It worked very well. We were able to knock out the PGC-1 alpha protein very well. And I want to use this Western blot to illustrate one key point about PGC-1 alpha in itself. I emphasized earlier that it's a very short-lived protein. It's there as a metabolic adaptation response to stress. So normally in most tissues, except muscle, PGC-1 alpha levels are very low, almost undetectable. You need to have some kind of stimulus or metabolic stress to see them. And then you can see in the knockout, we completely knocked out the protein. And then we also know now from the last 10 years of research that PGC-1 alpha is actually many proteins. So you see many bands on the blot, and we're able to knock out all of them. So like Charlotte Ling had shown in the earlier paper, in humans, we found also that low PGC-1 alpha in adult beta cells. So this was an inducible knockout that we knocked out in 2-month-old mice. So it wasn't low during development, but if you knock out in adult mice, they have lower insulin secretion. So now this is not glucose, this is insulin in response to an oral glucose challenge. So they had lower insulin secretion over time in response to glucose. We also did this as part of a hyperglycemic clamp in collaboration with the CRChin in Montreal with Thierry ARGCIA and Vincent Boitou. And they showed also what we had seen in our short-term test, is that overall insulin secretion in response to glucose is lower in these mice. They had lower C-peptide levels, which shows that it's, it probably is something that happens on a regular over time basis, but their insulin content of their beta cells was just fine. So it wasn't that they weren't expressing enough insulin, it's that they weren't responding to the glucose well enough. So because we're very focused, of course, on PGC-1 alpha as a model or regulator of mitochondria, one of the first things we did was look at mitochondria, because the easiest explanation, of course, was that the mitochondria inside the beta cells were not producing enough ATP, and you weren't getting enough stimulus to secrete the insulin in response to the glucose. So when we did a seahorse assay to look at respiratory capacity and response of these isolated islets from these mice, we were kind of surprised to see that there was absolutely no defect at all. And if you take muscle cells of a PGC-1 alpha knockout mouse and do the same thing, they have very significant reductions in oxidative capacity. Muscle rely on PGC for mitochondrial function, but the mouse beta cells didn't at all. Glucose worked just fine to induce oxygen consumption, and then coupled respiratory capacity was exactly the same, uncoupled. And ATP production, although slightly lower, it wasn't significant. So, it was surprising to us that these islets didn't need PGC-1 alpha or beta for their mitochondria to work properly in response to glucose. And it didn't explain why we were seeing reduced insulin secretion. So then, because the group down the road at the University of Montreal have focused a lot in recent years on the role of fatty acids potentiating glucose-stimulated insulin secretion. And we know that mitochondria are responsible for fatty acid metabolism as well as glucose. We always also investigated that. And we found, this is the interesting part of it, is that we found in response to palmitate, so you add glucose and palmitate together, fatty acids will potentiate the oxygen consumption in response to the glucose as well. So you'll see mitochondrial capacity go up. So this is in wild type islets. And then you see a greater coupled ECR. So the addition of fatty acids, not surprising, will ramp up mitochondrial metabolism to break down the fatty acid into energy. But in the beta cell knockout of the PGCs, this response was completely missing. So while they could respond to glucose, they could not respond to the additional stimulus of palmitate. They could not ramp up their mitochondrial metabolism. And coupled, when you looked at ATP synthesis coupled to this response, it was much lower. Although it didn't reach statistical significance, it was much lower than in the wild type mice. So they couldn't respond to the fatty acids as well. And this was illustrated, this could be illustrated very nicely actually in vivo. So if you isolate the primary islets, put them in culture, and then stimulate them with either glucose or glucose with palmitate, normally in wild type islets, you'll see a nice increase in insulin secretion in response to glucose at 16 millimolar. And then if you add glucose and palmitate together, this response will be potentiated. So you'll have higher insulin secretion when you have both the fatty acid and the glucose. But in the beta cell knockout, we had generally normal levels of insulin secreted in response to glucose alone, but this response was absent. You did not see the potentiation by the fatty acid. So this matched actually what we were seeing at the level of the mitochondria, is that for some reason now the insulin or the glucose stimulated insulin secretion was linked to this defective fatty acid response. We went a bit further because we wanted to know biologically actually how this affected the islets independence of just insulin secretion. So we looked at the islets themselves to see whether they were breaking down the fatty acids. So again, one of the functions of mitochondria is to be able to switch fuel sources from glucose to fatty acid if needed. And fatty acid oxidation is a major function. So whether they were able to oxidize these fatty acids that were coming into the islet. I don't show the data here, but we, in collaboration again with the CR Shum, we showed that actually fatty acid oxidation was quite normal. And despite the fact that fatty acid oxidation was quite normal, these islets still accumulated acylglycerol, so MAG, DAG, and triglyceride and cholesterol esterols. So most lipids were just higher in isolated islets. And it was known from many labs, including Mark Prenke's lab, that decreased lipolysis would lead to decreased GSIS because you are not producing some of these intermediates that are known to potentiate insulin secretion, so MAG in particular. So we wanted to understand why these fatty acids were accumulating a little bit. We knew that fatty acid oxidation at the level of mitochondria was fine, surprising, yet fine. So why were these fatty acids accumulating? Why were we not breaking them down into these intermediates? And so we dug a bit deeper and we ended up finding out that PGC-1 alpha and beta are actually important transcriptional regulators of the enzymes that are responsible for breaking down these triglycerides, ATGL and HSL. So you can see this, I don't show it, but it's at the mRNA level, as well as this is protein from isolated islets. So in conclusion, from this study, one, we were really surprised that PGC-1 alpha or PGC is not needed really at all to maintain mitochondrial, mass, function, basal mitochondrial function in islets. It's quite dispensable. But when it comes to the role of fatty acids in islets and their role in potentiating insulin secretion and what they're involved in terms of lipid metabolism, PGC-1 alpha actually seemed to be very important. And it was through these mechanisms that it was affecting insulin secretion, or insulin secretion in glucose metabolism. So that was published around 2015. So we were still kind of struggling a bit, because just to summarize where we are right now, we had looked at this model of mitochondrial inflexibility in muscle. We had put these mice also on a high-fat diet. The data I showed you already is mice that were on normal chow. But of course, we challenged them with this high-fat diet, and we had some very surprising results. When we challenged muscle-specific knockout mice, they were very insulin sensitive. So on the chow diet, they were insulin sensitive and glucose tolerant, and on the high-fat diet, it was even more. And they weren't more obese, so their fat mass was equal. So it seemed anyway in a young mouse that hadn't aged to 2 years old that loss of PGC-1 alpha, or loss of its role in mitochondrial flexibility didn't play a huge role, obviously, in the glucose intolerance and insulin resistance that is normally seen with diabetes associated with obesity. I just showed you a bunch of data to show that PGC-1 alpha and beta are important for insulin secretion in response to fatty acids. But if you give these mice a high-fat, high-sucrose diet, despite having altered insulin secretion, so that the insulin you saw earlier, it was measured during this GTT, and it is lower, but it doesn't correspond to problems with glucose tolerance, and these mice were equally obese at the same time. So the take-home message from this is that low PGC-1 alpha in these tissues in particular probably didn't make a huge contribution maybe to the pathogenesis of diabetes. But we had, over the years, collaborated with other people, and also in our own lab, taken a big interest in other tissues other than muscle and the beta cell, and this included the liver and fat. And so very early on, we had looked at fat, and for sure, mice with a fat-specific knockout of PGC are very insulin resistant and glucose intolerant on a high-fat diet. So the fat tissue plays a big role. These mice were not more obese, but they had problems with their adipose tissue energy metabolism. I'm not going to focus on fat today because we didn't end up going down that road, but we did end up going down the road of the liver. I became very interested in fatty liver disease at the time and its role in diabetes. And our data in the lab has suggested that even if we knock down PGC-1 alpha only 50%, so this is a het, we can cause problems with glucose regulation in the blood, and the mice ended up being a bit fatter or a bit more obese. So the liver and its, the ability of its mitochondria to adapt was starting to become more in the limelight, more interesting. And so we wanted to pursue this to see what role the liver had in regulating glucose homeostasis. And this is obviously not a mystery to most of this audience here, is how the liver is involved in regulating blood glucose levels. Of course, it's one of the major glucose sensing organs in the body. It's one of the first organs to see the glucose or the carbohydrates that we take in through our diet. It's also one of the first organs to see the insulin and glucagon that are secreted from the pancreas. It integrates all of these signals to kind of constantly balance the output of glucose, how much glucose it will produce to feed the rest of the body. In terms of fasting, you want to produce more glucose. Obviously, when you're fed, the liver needs to produce less. It's also seeing, insults from the gut, the guts, the intestinal tract. We know that the gut microbiome plays a huge role in these inflammatory signals. So it's kind of this organ that sits really at the center, in our mind anyway, of course, that of integrating all of these signals at once. It has to react very quickly. It needs to react to changes in metabolism, changes in energy source and environment very fast. So adaptability of mitochondria in the liver seem to probably be very important. We also know that in type 2 diabetes, the liver is very much important for the inappropriate glucose levels, high glucose levels in the blood, low response to insulin and insulin resistance in the liver plays a significant role in the disease, as well as at least 70 to 80% of people who are diabetic also have a fatty liver. So while they seem to be concurrent, they also likely play a role in crosstalk between each other. The liver and the pancreas are closely related anatomically as well as mechanistically. We are very interested in how they talk to each other. And so my interest in the role of PGC-1 alpha and metabolic inflexibility of mitochondria in the liver really actually stems back to one of the first projects I ever worked on, which was the question of how important is PGC-1 alpha in the liver to regulate gluconeogenesis. So, and was this gluconeogenesis involved in diabetes because PGC-1 alpha could drive too much gluconeogenesis or inappropriately. So what I show in this diagram here, just to catch you up to speed on some of the earlier work that originally formed my view of PGC, was, it was originally described in the liver as a factor that drove the expression of these enzymes, PepsiK glucose 6-phosphatase, to drive production of glucose in the bloodstream. So it was a master regulator of gluconeogenesis, and many people still describe it as this today. We knew that glucagon drove its expression. So when you're fasting, PGC-1 alpha levels are quite high in the liver, and that it was thought that this was responsible for turning on this pathway. And we knew that insulin very rapidly turned off PGC-1 alpha activity. So this was kind of the simple axis that we started from, and my job was to test whether this played a role in diabetes. The problem with that hypothesis is that would imply that PGC-1 alpha levels are high in diabetes, right? They're driving gluconeogenesis to make too much glucose. But there started to be more and more evidence, studies coming out, that wasn't actually the case in people with diabetes, in people with NAFLD, metabolic liver disease, if you're obese, so OBE is obese. Generally in the liver, PGC-1 alpha levels are quite low in this setting. And I alluded to that before, is that the more and more evidence that comes out, it shows that PGC-1 alpha levels in metabolic disease and diabetes is low. So this didn't really fit, with the hypothesis that PGC-1 alpha was inappropriately driving gluconeogenesis. And when we created our liver-specific knockdown or knockout, and the rest of my talk will kind of focus on this, well, not the rest, this data here will focus on this liver het model, because we had the most data in this model. So when you knock down PGC-1 alpha just in hepatocytes, our original hypothesis, if it fit with kind of the traditional view, was that we would lower gluconeogenesis by lowering PGC-1 alpha. So fasting glucose would be low. And we kind of saw that, right? We saw a non-significant reduction in fasting glucose levels in mice that had lower levels. But what we consistently saw when we did an oral glucose tolerance test is that post-prandial or post-glucose challenge, the levels of glucose in the blood were actually higher with lower levels of PGC-1 alpha in the liver. Going against our original hypothesis is that we should be able to reduce blood glucose levels by having lower PGC. So this was, interesting to us. Of course, we saw opposite of what we expected to see. We also saw higher levels of blood glucose after our meal. So this is after glucose, but if you just measure postprandial levels in mice, you can see they had the same levels. And this was concurrent with increased insulin, circulating insulin. So this is insulin during the OTT. So it wasn't that these mice were just not secreting enough insulin per se, right? They were secreting enough. They were actually secreting more than their wild type countermates, but they had more blood glucose. So this suggested to us, since the knockout was in the liver or hepatocytes specifically, that we had insulin resistance in the liver, that the liver was not responding appropriately to this high level of insulin that was circulating around. So I asked a very new PhD student in the lab who came in to kind of figure this out. It didn't make sense. It didn't fit our hypothesis. But we knew that PGC-1 alpha somehow had to be interacting with the insulin signaling pathway in the liver. So I asked, just look at the insulin signaling pathway and tell me whether having high levels of PGC or knocking out PGC has any effect on insulin signaling at the level of the hepatocytes. So in primary hepatocytes, either overexpressing PGC or knockout, he kept coming to me with data saying that it didn't matter whether you overexpress or knockout, you had increased phospho-AKT expression. So of course, this is data that no PI ever wants to see, because you have a knockout and a loss of function and a gain of function model that show completely the same thing. It's not very easy to explain. So it took a long time for us to get past this. And I admit that at one point, it wasn't something I wanted to really pursue. But that student kind of plugged forward and really took a tour de force and decided he was going to look at every aspect of insulin signaling he could find to try to narrow down why this was happening. Why do you have higher levels of phospho-AKT in both situations? And so I think a few years went by, and eventually he came to me and said, okay, I think I've figured it out. I was like, really? Okay. He's like really upstream. So if you're familiar with the insulin signaling pathway, the insulin receptor is autophosphorylated and then binds to one of at least 4 different proteins that are IRS1, 2, 3, 4, insulin receptor substrate. And so it's really early on in the insulin signaling pathway that this protein will bind, and then it is responsible for activating the downstream signaling pathways. And what he found was that when you overexpress PGC-1 alpha, you have some effect on insulin receptors itself. But what was most striking is that you see a decrease in IRS1 at the mRNA level and the protein level. So you can see it disappears. Whereas you have an increase in insulin receptor substrate 2, a significant increase. And you can see at the protein level, it's even more significant. So you really have an upregulation of IRS2 where you really tip the balance of the ratio of these 2 proteins in the cell. So, if you think, that the insulin receptor is either going to interact with IRS1 or IRS2, it would have more of a chance of interacting with IRS2. And then in the complete opposite model, in the loss of function model, you see the complete opposite. So when you have no PGC-1 alpha in the hepatocytes, you have an increase in IRS1. So IRS1 proteins are very significantly higher, and IRS2 at the mRNA level and the protein level decrease. So it's really a ratio thing. You're shifting the ratio depending on how much PGC you have there or not. And so that's depicting on what IRS protein the insulin receptor might be interacting with. And this would imply then that glucagon is regulating the IRS proteins. So a glucagon itself is having a direct effect on the insulin signaling pathway. So we looked at that and we saw an increase in, of course, PGC-1 alpha expression with glucagon, an increase in IRS1 and 2 expression. You can see that again, mRNA and protein level. What was interesting to us is that when we knocked down PGC, we really found that PGC was responsible only for the expression of IRS2. So it kind of it had an additional layer of specificity to regulate this IRS2 protein, and we found that that was through CREB. So this was a bit hard to wrap our head around again, because it suggested that glucagon signaling potentiates insulin signaling, which is not a concept that generally we think about when we think about, insulin and glucagon. They're generally counter-regulatory hormones, right? So I asked Aurel to test this in the lab and said, well, if this is actually true, then glucagon treatment in hepatocytes should stimulate AKT phosphorylation, right? And indeed it did, which surprised us a lot. But luckily at the time, the Habiger lab also showed the same thing. So a paper came out right around this time where they take primary hepatocytes or mice, and you can see that if you have glucagon and insulin at the same time, you have higher insulin receptor signaling. And you can see this very nicely in the mice, that mice who, this is an insulin tolerance test, they're normally given insulin, which lowers blood glucose. But if you give insulin and glucagon at the same time, your blood glucose levels will go lower. So really, insulin or glucagon can educate or influence insulin signaling in the liver. And so going back to the IRS1, IRS2 kind of story, what does that mean? Well, there was a lot of research before us, before we looked at this, which kind of gave us a hint as to what might be going on. And people had looked at what are the different physiological roles of these two proteins, right? Are they the same? Are they different? Are they really just, copies of each other? And it seems like they're not. They do share a lot of overlapping functions, but what does seem to be the case is that IRS1 will drive more of a program of anabolism, so lipogenesis, fatty acid storage, whereas IRS2 is more responsible for the inhibition of gluconeogenic side of insulin. So this would suggest that if IRS1 was higher, your liver would favor energy storage, and if IRS2 was higher, your liver would favor insulin to more be inhibiting glucose production. So this would mean, taking all of it together, that PGC1 alpha might be a way to educate the liver to have insulin work better or play more of a role on stopping gluconeogenesis as opposed to lipid storage. So really shifting the function of insulin. So we tested that first in vitro and then in vivo, so in primary hepatocytes. So on the y-axis, you'll see that this is the ability of insulin to shut off glucose production in hepatocytes. So normally if you take primary hepatocytes, you add insulin, you have a 40% reduction in gluconeogenesis. When you overexpress PGC1 alpha, you can enhance the action of insulin on its ability to inhibit it. So PGC1 alpha helps insulin to stop glucose production. And this was dependent on IRS2. So we knocked down IRS2 and the cells were no longer able to respond to the PGC. Cells that don't have PGCs in them at all are not able to respond to the insulin, so they cannot shut off glucose production. If you rescue PGC1 alpha, you restore that. And again, that restoration was dependent on IRS2. So in vitro, at least, we had pretty good evidence that this pathway, was there and that it could help to regulate the action of insulin. So this would suggest in vivo that, glucagon and PGC1 alpha are functioning in the liver to help insulin shut off gluconeogenesis. So if you increase levels of PGC1 alpha in the liver, you should be able to shut off gluconeogenesis more efficient. And that's actually what we saw. So we tailed-in injected mice with PGC1 alpha. We waited a week. We then did a pyruvate tolerance test, which is a substrate for gluconeogenesis. You can see that basally, if I went back to my original hypothesis, is that high levels of PGC1 alpha in the liver were originally thought to drive gluconeogenesis. We did not see that. So in fasting mice, we don't have high levels of glucose. But what we did see is that in response to the pyruvate, they were able to produce glucose just fine. But then during the time when insulin was released, they had lower levels of glucose in the blood. So we think that insulin is better able to shut off the gluconeogenesis because PGC1 alpha is telling the liver to shift the insulin signaling pathway to favor the brake on gluconeogenesis. So in essence, we kind of, we added to what was known about PGC1 alpha in the liver. We made the pathway a bit more complex and gave it a bit of depth. For sure, I might have frozen here. My computer froze for a second. I'm gonna wait. For sure, during fasting, glucagon comes in, drives PGC1 alpha to increase glucose production. That's true. We see this in fasting mice and primary hepatocytes. We know that this actually does happen and it agrees with what it's done. But we also think that at the same time, PGC1 alpha glucagon is coming in during fasting, driving gluconeogenesis, but then also priming the liver to respond to that soon to come insulin response. So PGC1 alpha through CREB is driving the signaling of insulin over to the IRS2 more dependent side, so that when you eat and when that initial insulin stimulus comes in, the liver favors shutting off glucose production. That's the first thing that it will do, is shut off glucose production. And then eventually insulin feeds back through AKT, inhibits PGC, that, an hour or two later, shuts this off, and then insulin can go over to the more anabolic functions, such as triglyceride synthesis in the liver. So some of the take-home messages I wanted to send really was that, in our studies, it made us look at insulin and glucagon again and not really just think of them as counter-regulatory hormones, is that they don't always oppose each other. That high PGC-1 alpha liver doesn't necessarily equal high blood glucose levels, which was, we think, a misconception that kind of has been propagated. And that there's really a lot of evidence now that low PGC-1 alpha in the liver is associated with metabolic disease in humans. So we're really trying to understand, what are the many aspects of like molecular mechanisms that could be involved in this. And we do think that some of the evidence that we've shown you is that it may contribute to poor glucose control and maybe eventually metabolic liver disease. And in the last 2 minutes, I just wanted to bring it back because I've shown a lot of data with loss of function models, particularly because we focused a lot on how we wanted to look at the dependence and PGC-1 alpha as a model of mitochondrial inflexibility. But I haven't done many transgenic models where we reverse or rescue it or look whether high PGC-1 alpha is beneficial, right? Looking the other side of the coin. We didn't make many transgenic models in the mice in the lab, but what we thought was very interesting was this SNP that I alluded to earlier that Charlotte Ling had shown was was associated with decreased insulin secretion. So there is more clinical papers out there that this SNP in PGC-1 alpha, which causes a glycine to a serine switch in the protein, is associated with insulin resistance, obesity, and decreased insulin secretion. About 50% of the population has the glycine form. So that's maybe a good thing if you believe the clinical data, because this is associated with lower inflammation and has a higher prevalence in elite athletes. So, we wanted to look at this model because we thought that maybe it was a more physiologically relevant model, and we knew that it was associated with diabetes. So one of the earlier master students in the lab, I asked them, can you figure out what this switch in the amino acid is doing? And what they did was they just overexpressed the protein and ran a Western blot. So at the mRNA level, it's equal, but they can see that the protein for the glycine containing variant is much higher. And other students went on to show that this is due to the glycine variant being just more stable at the protein level. So the mRNA level is completely the same, but the glycine protein sticks around longer. So its half-life is anywhere from 2 to 3 times longer than the serine. So since all the mice that we ever use, all the strains of mice we've looked are serine, and most species are actually serine. It's only really humans and some species of birds that have this glycine variant. We decided to switch the variant and switch the serine version in mice to the glycine. And what we found was that this did what it did in humans, actually, is that it will stabilize the mouse PGC-1 alpha protein. So we'll have more PGC-1 alpha around in the mice. And so we created a mouse model of this variant, which we think is a more physiologically relevant gain of function model per se than doing a transgenic in this situation anyway. And we're really interested to characterize this mice more. And I wish that we had more data at this point, but I don't have any to show that, but it's something you can look forward to. So in conclusion, and hopefully there'll be enough time for questions, I welcome them all. The last almost 10 years, we've really been interested in this kind of central role of mitochondria and how they're able to adapt to all of the different metabolic stimulus or challenges that we see in life, aging, lifestyle, diet, and how all of the organ systems kind of talk to each other and each play their own individual role. We know that low PGC-1 alpha is generally associated with metabolic disease, and we can model that in the lab and try to figure out why. We know that it plays different roles in different tissues, and it's been a really interesting journey to figure out what those have been, and that it's a really important regulator of metabolic flexibility. We also know that low PGC-1 alpha doesn't cause diabetes, but if you have low PGC-1 alpha, you have reduced efficiency of energy metabolism, and that, when combined with aging, diet, or other factors, can possibly worsen diabetes outcomes. So it's a modulating factor in this process. And I'm just gonna, again, thank everybody who contributed this work. it's really been an interesting 10 years that has gone by very, very fast. Thanks. Oh, okay. I see there's questions now. So I'm gonna open them up. Okay. Jim. Congrats on an amazing career so far. Oh, thanks. Much more. What do you think about the possibility that other tissues in glucose homeostasis are modulated by lipid spillover? Oh, that for sure. I don't think those studies have been done yet, like lipidomics in the blood per se. I think the more and more studies are happening that are showing that many things are dysregulated in terms of what's being secreted from the muscle, but probably also the liver, other tissues, in response to this kind of lower PGC or metabolic inflexibility, as I like to think of it. So I think the more omics that are done will help us kind of define that and what different species are being regulated. If they're done at the end, okay. Oh yeah, sorry, the question, I assume that everybody can see it, but is the effect of glucagon on insulin signaling dose dependent on the effects linear? It's not something we looked at. Actually, we did, most of our studies were done at a 25 nanomolar dose or a 50 nanomolar dose. We didn't look at the effects dose dependent. I was wondering if you have looked in the adipocyte PGC-1 alpha deletion and the possible effect on adipose. No, we didn't go back. So the original knockout we did in the fat was done while I was a postdoc, and we haven't gone back to that mouse since then. So it would be interesting to look at insulin signaling in that mouse. Well, it also could be something that we look at in the new model, the CRISPR-in mutation that we have now, where we have more stable PGC, to see if we see effects on insulin signaling in the adipose. It's a very good idea. Are there sex-specific effects of PGC? Oh, yes. So I didn't go into it today, but we've looked at it that extensively in the liver, and it's what looking at both males and females made us realize that females, one, have higher levels of PGC-1 alpha in livers, about 4 times higher levels of PGC-1 alpha in the liver. We've looked briefly in other tissues. It's true in other tissues. We know from other people in the field who have looked. So I think females have higher levels of PGC. We think this is through estrogen. So we are able to show in the liver that estrogen potentiates PGC-1 alpha expression, and that some of the beneficial effects of estrogen in liver are dependent on PGC. So there definitely, if PGC sits downstream of estrogen, it's important in other tissues, I think we're going to continue to see sex effects on this. And we in every other experiment we do after that, now we look at both. So I anticipate we're going to see more. As PGC-1 alpha interacts with PPARs, do you think the effect on insulin signaling could be? Definitely, yeah. So we have a collaboration with Hervé sorry, I'm losing my train of thought because I've been talking too much, in Toulouse, and Guy in Toulouse. And he is focuses a lot on PPAR alpha signaling, and there's a lot of data, there might be some overlap. I mean, we know that PPAR gamma and PPAR alpha are major targets of PGC. It's one of the first transcription factors that were shown to interact. Given what we know about PPAR alpha's role in mitochondrial function and fatty acid oxidation in the liver, it's a very interesting question to figure out whether PGC-1 alpha is responsible for, in part or a lot, of what PPAR alpha does. Yeah, sorry. Yeah, I realize it's hard that you guys can't see the questions at all. Okay, and you can't answer me, so I'll try to be more precise in my reading of the questions. So, may there be a link to ERR gamma, as shown by the Evans group? ER gamma potentiates mitochondrial function. Yes. So the ERRs are also one of the major families of transcription factors that interact with PGC-1 alpha coactivators. Much of the function of PGC-1 alpha is via ER alpha. Not as much has been done to look at ERR gamma. So it would be interesting to see if ER gamma also plays a role in the downstream pathways. But in general, PGC-1 alpha interacts with at least 20 different transcription factors. So teasing out the different roles of each of them is difficult, particularly in the models we use. But combining some of the different strategies to rescue or knock out could identify specific pathways. All right.
All right, looks like that is all the questions we have. Maybe one more popped up. Do you want to? We'll have this one be the last one.
Okay. Have you looked at PGC's role in what are SC beta cells? No. I probably shouldn't. Oh, stem cell derived. Okay. We haven't, but we have yes, we haven't looked at it. There is one paper from 2014 from Valtet and Blondeau's group where they overexpress PGC-1 alpha in vivo in beta cells. Like, so if it's there during development, high levels, it interferes with beta cell function. So we know that they don't screen as much insulin, but that's all we know. So whether that's involved in maturation and identity of the beta cell, I think it's still a debate. There are different expressions of some of the key factors that kind of define a mature beta cell or not. So we have a collaboration ongoing now with Francis Lin to look at this in iPSCs and to kind of identify what PGC-1 alpha expression levels or different proteins within that family, how they might play a role in the identity of a beta cell. Yeah, but I mean, to answer the question kind of bluntly, we can do a full knockout of PGC-1 alpha and beta and a whole body knockout of these things, and the mice are alive and their islets kind of look normal and they have normal architecture. So in general, I would say that they're probably not essential or playing a major role, but it might help to nuance, I guess, the system. More would need to be known.
Okay, great, thanks. I will end the presentation there. I would just like to let folks know that we have some other webinars coming up. So tomorrow, actually at noon, the award recipient of the Lifetime Achievement Award, Dr. Patricia Brubaker, will be presenting on the glucagon-like peptides. So that's tomorrow at noon. On Friday, November 27th, Dr. Alice Chang will be giving an update on chronic kidney disease and diabetes. So a couple of very exciting, those will probably be our last two webinars of the year, so you'll want to check them out. All of our webinars are free, but if you found today's presentation useful, you may be interested in becoming a professional member with Diabetes Canada. You'd get access to the Canadian Journal of Diabetes, the Diabetes Communicator, all the archive versions of those, as well as other member benefits. So you can head to diabetes.ca/healthcareproviders/professional membership to join. And again, this webinar was free. So Diabetes Canada could certainly use your financial support more than ever. COVID-19 has hit us pretty hard. So www.diabetes.ca/donate. So with that, I would like to thank Dr. Estell for presenting today and congratulate her once again on being the 2020 Young Scientist Award winner. I'll give you the last word, Dr. Estell.
I just would like to thank everybody for tuning in and listening, and really for the support of everybody over the years. It's really, it wasn't just me for sure. It's really taken the community, the islet community, the diabetes community in Canada has really been supportive of us in my lab and my trainees. So it's how we've gotten to where we are now. So thank you, everyone.
All right, have a great day.
Bye.